Hi there, and welcome to a video about viral structure. So I'm just going to go through a few of the basics of the basic components of viral structure, so the two basic components that are there, and then I'll talk a little bit about some optional molecules / structures. So viruses are made of two very simple parts. There's the capsid, which is a protein coat, and it's made up of something called capsomeres. Those are the subunits. So we could say made of capsomere subunits. So the little protein parts put together to make this coating. And that's the coating on the outside of the virus. There can be additional coatings, which I'll go into here in just a second. The other component of viruses are nucleic acids, or a nucleic acid. And I said acids because there's different possibilities. So that's in the interior, and it's what we can call the core. So inside the core, and it's one of four types. So we typically think of DNA or RNA, we think of one as double-stranded and one as single-stranded. However, in viruses, you can have double-stranded DNA, and this is or. You're going to have one type. Single-stranded DNA, or double-stranded RNA, so that's quite a bit different than what we're used to, or single-stranded RNA. So you have one of those four types. Okay, so what else can you have? Well, you can have some optional items. So this is all viruses. So some optional add-ons. There is envelope, which is a phospholipid bilayer. Followed by spikes, that's another option. And those are typically protein-based, so protein or lipoprotein, protein projections. And they just like what they sound like by the name. Other enzymes, such as, and these are enzymes that are unique to viruses, so we don't have those. So normally the viruses always use our cellular machinery, our enzymes, our cellular structures, but there's some enzymes they need that we don't have. So those may come in with them. So one is reverse transcriptase. That's seen in viruses such as human immunodeficiency virus. And it's used to kind of reverse engineer or reverse synthesize RNA back to DNA. So we don't go that direction, but viruses can, these particular types. You can also have polymerases. And then you might have other protein types. So there are appendages. So in one complex type of virus that's fairly unique because it does have all these appendages, there's things like tail fibers and a sheath. So you can have that. You can have M protein, which is a coat inside the envelope of some viruses. And again, these are all options. Lipoproteins. And again, these are just exceptions at certain times. And then other fiber-like proteins as well. The size of viruses, they're what's called ultra microscopic, meaning that you can't see them, most viruses, under a typical light microscope. So they're in the nanometer range, so somewhere around 20 to about 200 nanometers. There are exceptions to that because this is biology, so we always have exceptions. There are some as large as 1,000. But we're still done in that nanometer range. so keep that in mind. So you would always need an electron microscope to view these. And another key feature of viruses is that they are what's called obligate parasites, meaning they cannot, basically they exist, but they cannot reproduce and do any of the functions that viruses need to do to make more copies of themselves without a host. So they actually need the host cell machinery. They are also what's called acellular, which relates to that, meaning they do not have a cell structure. So these are the two parts. So capsid, nucleic acid. So there isn't a cell membrane. However, they can pinch off some from the host sometimes. No nucleus, no nucleoid region, no ribosomes. They don't have all of those cellular structures. Certainly wouldn't have anything like organelles, cytoplasm. So they're just very, very basic, but that simplicity sort of gives them a certain elegance in terms of difficulty to treat, because they are so simplistic in terms of the structures that are in each virus, that it's difficult to make drugs that target one of these without impacting the host. And it makes them very, very quick to replicate as well. So quickly to review, all viruses have a capsid and some type of nucleic acid. It's one of 4 types, not a combination. And then there are some viruses that have additional molecules, so they may have a lipid bilayer they take from the host. And then they may synthesize other proteins and enzymes, including spikes, specialized enzymes, and then some protective or structural type proteins as well. And their size is very, very small, so they're not able to be viewed with your normal laboratory compound light microscope, but if your normal laboratory has an electron microscope, that's what you would need. So electron microscope is needed to view most of those. There are a few that are very large, but most of them fall in this pretty small 200 to 20 nanometer range. They cannot multiply or make more copies of themselves outside of the host, so they are obligate parasites. And they're basically inert until they get into the host, sort of inactive, and they're acellular. There is some debate about whether they're living or not, because they do have these sort of certainly active functions once they get into the host cell, but they're not able to perform any of those outside of the host cell. Plus they lack a cell structure, so they don't really abide by that cell theory as a basic unit of life. Okay, that's what I have for viral structure. In other videos, I'll go over more details on the layout of some of these components. I thank you very much.